Okay, HL, grade 12 biology, here is your lesson for meiosis. These are just the things that are only relevant for you guys, building off of what we did with the SL students. So this is subtopic 10.1, meiosis, and this is our list for today. Chromosomes replicate in interphase before meiosis. Crossing over is the exchange of DNA material between non-sister homologous chromatids. Crossing over produces new combinations of alleles on the chromosomes of the haploid cells. Chiasmata formation between non-sister chromatids can result in exchange of alleles. Homologous chromosomes separate in meiosis 1. Sister chromatids separate in meiosis 2. Independent assortment of genes due to random orientation of pairs of homologous chromosomes in meiosis 1. And our skill is drawing diagrams that show chiasmata formed by crossing over. This is nothing completely new. This is all just building more detail off what we did with SLs in topic 3. So here's our target for today. I can analyze the process of crossing over and independent assortment, and I can clarify the difference between chromosomes and chromatids. So I'll start by talking about meiosis and interphase, specifically in meiosis, of course. So what is the purpose of interphase? Why do we do interphase? Well, we already talked about dividing interphase into 3 main parts. We have G1, S, and G2, called growth 1, synthesis, growth 2, where we're doubling the cytoplasm and the various organelles inside of it. Then in synthesis, we're copying the DNA. And then in G2, we're double checking all those things, making sure we're ready for division for actual meiosis here. Meiosis 1, meiosis 2, cytokinesis. So the purpose is to copy all the DNA and grow the cell to be able to divide. Why doesn't it happen twice? Other phases of meiosis happen 2 times, right? We have prophase, metaphase, anaphase, telophase 1, prophase, anaphase, metaphase, telophase 2, but we only have 1 interphase. Why is that? Remember, the goal of meiosis is to produce haploid cells. So if we start off with 2 copies and we double it to having 4 copies in 2 pairs of chromatids into that's chromosomes for 4 separate chromatids, and then divide it once. We're going to put 2 chromatids in the first division daughter cells. If we then copy it again, we're going to end up with 2 chromatids in the second division too. The goal is to get to 1 chromatid only to get to haploid. So we only double it once here, and then we divide it and divide it again. If interphase didn't happen, how would that affect meiosis? What would meiosis be different? Well, if our cells didn't get bigger and copy the DNA, we could divide them in half and we get 2 haploid cells, but not 4. We wouldn't have all the crossing over and all the possibilities there as well, right? That all happens getting ready to copy the DNA. So we need to get bigger and we need to copy DNA to be able to mix and match 4 chromatids. Okay, let's more talk about interphase here. So in interphase, what's happening is DNA replication. We're going from a single copy of the DNA, we're unzipping it, and we're copying it to make 2 new copies. Okay, and this is going from a diploid cell with 1 chromatid per chromosome to a diploid cell with 2 chromatids per chromosome. They're still called diploid, and they're still called chromosomes. So here I have diploid cells with 1 chromatid and 1 chromosome. This is still called 1 chromosome, but now they have 2 chromatids. So diploid, diploid, same thing. It's confusing, but it still means diploid. So the clarification here is how many chromatids make up a chromosome. It makes a difference between 4 chromatids preparing for meiosis and to make 4 haploid cells, or 4 chromatids for mitosis, dividing once to make 2 specific chromatids, 2 chromosomes. We will go through into detail of how this whole process occurs in topic 2.7, which is replication, transcription, and translation. The replication part is this. We'll be doing some of it with SL in 2.7, as well as adding more into it with topic 7 for you guys for HLs. Okay, so chromatids. I want you guys to watch this animation. Hopefully it works on the screencast. And take a look and make note of when the homologous chromosomes start. Remember, homologous chromosomes means the same chromosome. So chromosome number 2. There's 2 homologous chromosomes of that one. When they separate and when sister chromatids separate. Let's see if this works here.
Meiosis follows interphase after the chromosomes of a diploid cell have replicated. DNA replication occurs during S phase. After the 2 successive cell divisions of meiosis, a single cell will produce 4 cells, each having the haploid number of chromosomes. Prophase 1 of
I will
meiosis
link
1
this
begins with
animation
the condensation of
because I have a feeling you
chromosomes
can't hear
and the
it.
vesiculation of the nuclear membrane.
Maybe you can.
Centrosomes, which duplicate at the beginning of meiosis, begin moving apart, and the spindle fibers begin forming. Early in prophase, synaptonemal complexes form, and the homologous chromosomes are seen to align next to each other to form a structure called a bivalent. A bivalent is composed of 4 sister chromatids. Crossing will occasionally occur between homologous chromatids within a bivalent. During prometaphase 1, the spindle apparatus continues to form and bivalents become attached to kinetochore microtubules. One pair of sister chromatids is connected via kinetochore microtubules to one pole, while the homologous pair of sister chromatids is connected to the opposite pole. The complete assembly of the spindle apparatus occurs during prometaphase 1. The chromosomes align at the metaphase plate during metaphase 1. It is important to notice that bivalents are aligned along the metaphase plate rather than individual pairs of sister chromatids, as in mitosis. Anaphase 1 involves the separation or disjunction of homologous chromosomes, with each pair of sister chromatids moving to the opposite pole of the cell as the kinetochore microtubules shorten. When the conjoined sister chromatids reach the poles of the cell, they detach from the spindle apparatus, and the nuclear membrane begins to reform. A cleavage furrow forms, and cytokinesis ultimately results in 2 cells, each having the haploid chromosome number, with each chromosome consisting of a pair of sister chromatids. This completes meiosis 1, and after a period of interkinesis, the 2 daughter cells start the second cell division cycle called meiosis 2.
Okay, so homologous chromosomes separate in anaphase of meiosis 1. So anaphase when you're seeing here. And then sister chromatids during anaphase of meiosis 2. So both these divisions are happening in anaphase when chromosomes are being pulled apart. Homologous chromosomes anaphase 1, sister chromatids anaphase 2. Why do we have to divide twice? Why do we have to divide both times? Because our goal is haploid with a single sister chromatid. We want only one chromatid to make sperm or eggs that are haploid, and they can come together to make a diploid zygote. Okay, so we have these sister chromatids, homologs here that are going to be together after being replicated to produce the sister chromatids. And these will separate, 2 of them in meiosis 1, and then the 2 halves, the 2 chromatids, will separate in meiosis 2. Okay, so specifically how crossing over happens, the loving to with this in the HL is with Chiasmata. So the purpose of crossing over is what? Why do we bother with crossing over? Well, it's an excellent way to increase genetic variation. We have more variation because we're not just having alleles from mom and dad, we're having an allele, chromosome from mom that has a bit of dad, and vice versa. So it gives even more variation. Now, what is a parental chromosome and what is a recombinant chromosome? So the parental chromosomes here, you can see the ones on the end that's just the dark blue and just the light blue. Those are the ones that there was no crossing over that happened. They are still intact by themselves. And the recombinants are the ones that have a bit of the parent here, the other parent, and then they have some themselves. So the chromatid that has crossed over and has bits from each, that's a recombinant. And the parental is the normal. So we're going to have to talk about these later when we talk about linked genes later on. So those terms are important to clarify. So which chromatids did Chiasmata form between? Which of those 4 chromatids in a pair of homologous chromosomes is going to happen between? It has to happen between our non-sister chromatids, the ones from the 2 different chromosomes, from the parental and the maternal. If it was from the sister chromatids of the same chromosome, then nothing would get mixed around, right? Could be the same DNA. If the parent is homozygous for a trait, the same DNA, nothing's going to happen. We're not going to increase any variation, putting all the energy for no good reason. Now, if non-homologous chromosomes crossed over, if we had like this is chromosome 3 and this is chromosome 2, they have different genes on them. If that crossing over happened, that could be a disaster. It could create a translocation mutation where chunks of DNA are missing from chromosomes or extra chunks are at the wrong spot. It's a very, very bad thing. So it's really important crossing over is controlled and done in the right way. Now the skill of the IP wants you guys to draw diagrams to show chiasmata formed during crossing over. So 2 diagrams here. This is a light microscope diagram and this is an electron microscope. Obviously this one is much more detailed. Choose one to draw, pause the video and draw it. I recommend using color to help tell the 4 chromatids apart. Otherwise it's really easy to get them mixed up. So pause the video, do your drawings. Make sure you label them as well. Which chromatid is which one? Which chromosome is which one? Okay, here's our key ideas in the first part of the lesson. Interphase takes a diploid cell with a single chromatid to a diploid cell with a pair of sister chromatids to prepare for the 2 divisions of meiosis 1 and meiosis 2. Crossing over occurs between non-sister chromatids of homologous chromosomes at chiasmata, and crossing over increased genetic variation through a mixing of the parental chromatids into some recombinants. Remember, key ideas are a great moment to check for understanding, ask any questions you may have. And I'm not there today. If you have questions, send me an email or make a note to ask me next class. Either one is fine. Okay, let's talk about Gregory Mendel. So we're going to be talking about him more in our next unit after mocks in January. We're going to start connecting with things here. So Gregory Mendel is often described, he's the father of genetics. We learned about him in the 10th grade if you were at RAS. And there's 3 main laws that he wrote about inheritance patterns in genetics. He wrote the principle of dominance, he wrote the law of independent assortment, and he wrote the law of segregation. He's famous for doing all these crosses with sweet peas. So here's a cross with peas where he's looking at the color of the pea pod and whether the pea pod is constricted or smooth, like wrinkled or smooth pea pods. So Mendel did this before we knew what DNA was, before we knew what meiosis was, any of those kinds of things. But 2 of the laws he created that are based on patterns he noticed in collecting thousands of pieces of data and evidence from all these crosses of pea plants he did in his garden for many, many, many years are caused by meiosis. So which of those 3 laws are caused by meiosis? Well, the first one is definitely law of independent assortment. I've already mentioned this, that the chromosomes assort independently at the metaphase plate. They line up independently, regardless of each other. It's not all from dad on the left, all from mom on the right. It's completely random where they line up. That's independent of each other, independent assortment. And then segregation is also from meiosis. It means which chromosomes end up in which gametes. That's completely, again, the segregation and separation of them is the cause of meiosis. And Mendel figured these out just from inheritance patterns and kind of logicking his way backwards based on tons and tons and tons of data. It's very impressive. Here's another diagram to help with this. So we have segregation. We look at meiosis. Here's the top here. We have our diploid cells. So pairs of alleles are separated. We make gametes. We're separating and segregating our alleles. And each gamete gets 1 copy of each one. So they're haploid. And it's independent. They are sorted independently, right? So the pairs of alleles are sorted and moved into the different cells, different daughter cells, but it's completely random. There's no plan of how it happens. And then dominance, that one allele dominates the other. So, for example, purple dominates white, and short is recessive, but tall is dominant. And we'll learn more about this in our upcoming unit in the new year, or review it if you learned it before. These 2 ones are all about meiosis, and this one is not meiosis, but we will talk about it more later on. Okay, so let's connect meiosis to reproduction. Thinking about 11.4, way back when, can you explain how this topic, 10.1, is connected to 11.4? Well, meiosis is how we do the genetic material part of both oogenesis and spermatogenesis. So we were focusing on the other parts of this in 11.4 about adding the acrosome and the tail to a sperm, getting rid of polar bodies to get a very large ovum, all those kinds of things. We focus on that 11.4. Meiosis is what's happening in the nucleus, in the DNA part, which is equally, if not more, important. Okay, remember the other things that are necessary to make a sperm and eggs or oocytes is what we're going to be doing, getting rid of 3 polar bodies to make an ovum, to make oocyte. We're going to add an acrosome head. We're going to add the flagella, all that kind of stuff to sperm to be able to make that able to have motility, all those kinds of things. So it's a connection here between 11.4 and the process of making the sperm and eggs, and then meiosis, the actual genetic component of making sperm and eggs. Okay, so here are your key ideas. Mendel's law of independent assortment and his law of segregation can be explained by meiosis. Gametogenesis and oogenesis both require meiosis to produce the correct amount of genetic material for the respective gametes, although both require the formation of additional structures on the final oocyte in the spermatozoa. That is the end of the lesson for today. I do have some practice cards. There are 24 cards. If Miss Couture can't find the cards in the filing cabinet because I forgot to pull them out, I'll also post the file for them on Schoology and practice. Good self-assessment to practice. And remember the to know list from subtopic 3.3. Next lesson is on subject 9.4, so make sure you read that for our lesson on Tuesday. And as always, any questions, things, feel free to send me an email or come see me on Tuesday. Or Monday. Have a great weekend, guys. Take care.